Hello. We can go ahead and get started. My name is Todd Haim, and I represent the National Institute on Aging, and specifically our small business programs today, which is within the Division of Extramural Activities. We are joined by colleagues at both NIA and NINDS across different programs and scientific divisions. So I will ask each panelist to introduce themselves. First, Nigel Greig.
Thanks very much, Todd. My name's Nigel Greig. I'm a tenured scientist at the NIA within the Intramural program, which does the internal component of the research associated with NIA.
And next, Ron.
Thanks, Todd. I'm Ron Kohanski. I'm the director of the Division of Aging Biology. Our major emphasis is basic biology and mechanisms of aging.
Great. Suzana.
Thank you, Todd. Good morning, everyone. Suzana Petanceska here. I'm a lead at the Office for Strategic Development and Partnerships in NIA's Division of Neuroscience, and I'm program director in the Translational Research Branch, overseeing the front-end target discovery and validation efforts.
Thank you. And Chuck?
Hi, I'm Chuck Cywin. I'm a program director in the National Institute of Neurological Disorders and Stroke. I have responsibility for the Blueprint Neurotherapeutics Network, as well as the analgesic therapeutic development program as part of HEAL.
Great. And finally, last but not least, Nick.
Hi, Nick Langhals. I'm a program director at NINDS as well in the Division of Translational Research. I oversee our translational devices programs that are focused on moving technologies from late stage development into first in human applications. And I also wear a hat as a team lead within the BRAIN Initiative, overseeing the tool, one of the tools and technologies teams there, as well as several associated funding opportunities.
Great. So I think it's already clear that we'll be discussing a variety of different and exciting scientific initiatives and activities across the two institutes. The way the format will work is we'll have each of the speakers talk a little bit about what they're specifically focused on, and then we will save time for Q&A. Please use the Q&A button at the bottom of the screen to send questions. We really do want to be able to take your questions, so please send questions as you come up with them. We'll hold all questions to the end, but definitely plan on getting to them. So without further ado, Nigel, please go ahead.
Great, thanks very much. So thank you very much indeed. So the Intramural Research Program of NIA covers a broad area of sciences that's shown two down here. A whole series of disciplines, both from the basic and clinical point of view. It's set across a number of campuses in Maryland, and basically the program, which has about 400 scientists, or more than 400, seeks to understand changes associated with healthy aging and to find criteria for evaluating when changes become pathologic, and to mitigate that through new drugs, new devices, etc. So there's a common interest in diseases associated with aging, such as Alzheimer's, Parkinson's disease, etc., shown here at the bottom, but also in the determinants of healthy aging are also being defined to improve longevity. Next. So these 400 scientists across discipline have a very healthy interest under the leadership of Luigi Ferrucci, who's the scientific director, to focus these towards translation. And there are many areas of potential collaboration that we can interact with you. Some of that involves the technology that's developed within the intramural program, and some of that undoubtedly relates to technology that can come from you. So we have common interests across all these. So what I'll focus on is some of the technology that comes from us, and then you can come with us, you can come to us with your technologies. If one goes to the NIH OTT website, one will pull up this window here, and one can input and search for various technologies to see which institutes across the NIH have interests, and all the institutes are represented from the technologies that are available and can be licensed out. And we can see one of the NIA technologies at the bottom here. The contact person for NIA is Nikki Guyton, with the email address shown here and her telephone number. Next. So an article from Forbes in 2018 was focused on what's the best predictor for scientific success. And out of 8 elements that they described, they said that one of the best predictors for future success was past success. So let's look at the success for some of the technologies that have derived from NIA, but also from other institutions within the NIH. We can see that number 1 ranked for many, many years was a device that came out of the cardiovascular program of NIA, which is still run by Ed Carter, and with Taxol-coated stents here. They're used in angioplasty to treat atherosclerosis, for plaques in major blood vessels. One stents them, but there's often regrowth across these stents, and that restenosis is largely mitigated by Taxol covering. And it's hugely successful, both commercially as well as for the treatment in the public good. Next. Another success that came out of Josef Piether when he was at the NIH, and for numerous years was one of the greatest successes of NIH technology, was the development of cyclodextrins for formulating drugs. So these cyclodextrins have a lipophilic core where you can hide a lipophilic drug, but the outside is very water soluble, allowing you to put the drug in there so it can come out and have better bioavailability. Next. So from my own research group, for current collaborations that we have with industry, just a few examples. GLP-1 receptor agonists, or incretin mimetics, are widely used and approved for the treatment of type 2 diabetes, and we were the first group to repurpose them towards neurodegenerative disorders. So in a publication in Lancet, demonstrated with Tom Fultini from University College London that they were effective in a phase 2 clinical trial for Parkinson's disease. So Tom has a phase 3 clinical trial. There are multiple other clinical trials for a whole series of disorders, and there's lots of preclinical support for a whole variety of disorders. Patents got licensed out to Peptron, a South Korean company, that are now in clinical trials. Those clinical trials are being supported in large part by NIA, and samples will come in for blinded analyses to NIA. Next. Another company, Anovis Bio, is a small Alzheimer's, Parkinson's disease focused company. I pulled this from their website, and you can see a number of agents in clinical trials. All these agents are actually NIA intramural research developed drugs from my program. And so it demonstrates that companies can have underpinning technology from NIA. Next. The same with Aristea Translational Medicine. Clinical trials are about to start in Alzheimer's and TBI on Fenzarin, which is another NIA-developed drug, originally from my program that started this company. Next. And a very recent one, Avis Bio, is a company started in South Korea as well as Maryland. Its founding technology are pomalidomide analogs, also from NIA, towards neurodegenerative disorders to mitigate neuroinflammation. Next. A very exciting area from a colleague of mine, Ruin Modell within NIA, across collaboration with Carlos Zarate, NIMH, NCAT, Craig Thomas, Todd Gould from University of Maryland, is the development of ketamine to treat resistant depression. It's immediately effective. However, there are some liabilities, but those liabilities can potentially be mitigated by the use of a specific metabolite, which is this 2R6R hydroxynorketamine, which is being developed into the clinic and will be developed into phase 2 clinical trials and then potentially licensed out. So another area of potential collaboration and technology. Next. So we have within the intramural program of NIA, but many other NIH institutions, a huge amount of translational research going from bench to bedside continuously with huge numbers of collaborative scientists. There are multiple mechanisms to support these collaborations. We have experts here that you're going to hear from that can tell you what grant resources, what supporting expertise. This is a win-win situation. It's a win-win for us because our technology moves forward towards public good. It's a win-win for you, and it's a win-win for the US public. We need to collaborate. Thank you.
Great. And next, Ron Kohanski.
Thank you very much, Todd. And it's good to follow Nigel's excellent presentation. I hope I can do as well. Next slide, please. I represent the Division of Aging Biology, which is focused primarily on three aspects of the rate of aging, which can be broken down into basic, applied, and translational research. We have 4 sections within the division where we, in the genetics and cell biology branch shown in the upper left corner, looking at the interactions among the hallmarks of aging. Hallmarks of aging, generally speaking, are areas where biology that has been functioning well functions less well, and the result of that is a decline of capacity, a decline of resilience, and an increase in frailty in older animals, including older humans. And in those metrics for the hallmarks of aging, some of the outcomes are shown in the 2 figures in the middle. One of them is from Belsky, and it represents how populations can be stratified within a longitudinal study, but stratified across a single age. So this is basically a distribution of people who are 38 years old, and some of them seem to be aging faster than others, and some more slowly than others. So within any of these populations, you can find vast differences in how they age, and that is also a way of breaking out a population to look for where drugs might be effective, both in the laboratory and in human populations, since not everybody responds to the same set of drugs with equal efficacy. And in the figure to the right of that shows the heterogeneity in the rates of aging, which is linked, the 2 figures are linked in the concept that aging is specific to the individual, and within an individual, specific to their organs and cells. Not everything ages the same way in one person, and not every person ages the same way as others. However, the other important point from this work from Michael Snyder and Brené's labs is that you don't have to wait 40 years to see what the rate of aging looks like. And that means that you can test your interventions as they are developed, interventions against aging, over shorter time periods, as short as 2 to 3 years, which is the time frame here for these results. And this is a mixture of molecular and clinical diagnostics. In the aging physiology branch, there we look at our investigators that we support, look at the relationships between the hallmarks of aging and specific aging phenotypes. And those findings actually can be the basis for phenotypic screens, in addition to screens that are designed to target specific molecular pathways. In the biological resources branch, we provide animals across their lifespans, primarily rodents, but also some non-human primates, access to the non-human primates, as well as databases. And they are looking at programs of resilience related to the hallmarks of aging. By resilience, what we mean is if you perturb the animal, how well can they react to that perturbation? And that resilience does decline with age. And then in the office of the division director, we are developing an emphasis on aging phenotypes and human health. Next slide, please. So what is contained in each hallmark of aging? And the point of this slide, I can tell you at the beginning, is what is the appropriate level of granularity when examining a hallmark of aging and when testing these hallmarks for potential interventions. So going from left to right, the buckyball type diagram lists several of the hallmarks of aging. Proteostasis is one that's been expanded to look at what happens inside the cell. Various organelles are involved in proteostasis, and those organelles can be visualized, if you will, and they themselves can be used for screening of compounds. And then underneath those are, of course, the molecular networks that lead to, through the functions of organelles, lead to the proteostasis dysfunction as indicative of aging. Obviously, you can press on any of those nodes as levers in the molecular pathways and have large effects, or you can press on selected of those molecular entities that make up the network and have perhaps less dramatic effects. And it also suggests ways in which combination therapies would be useful. Next slide, please. So what we've done in this is to ask, if you go to the clinic and someone is, your physician is examining you, this is baseline data for how well are you aging. And what it does is it lays out a number of things that a physician might sample, some of which they do sample regularly, such as metabolites in the blood. But you can also look at the cells that are in the blood or cells that are from various tissues, and you can find out things about microbiome, epigenome. All these various hallmarks of aging can be related to what can be learned in the clinic. In addition, some biopsies are reasonable, such as fat biopsies or muscle biopsies if you get consent. Others are done in relationship to a disease, but within those disease biopsies, you also have unperturbed, relatively unperturbed tissue and cells. And that leads to the issue of cell heterogeneity, which leads back to the issue of aging is heterogeneous. So what we hope to see is linkages between the clinical analyses and the molecular underlying basis. And I believe I will conclude with that. Thank you.
Thank you very much. Very exciting, and I look forward to seeing how this evolves over time. So next we have Suzana.
Thank you, Todd, and thank you, Ron, for that beautiful staging for the next presentation. So Alex, if we can go to slide 4. Thank you. So one of the major components of our Alzheimer's translational research program are the enabling infrastructure programs that are built around open science and open source principles. And collectively, these programs are delivering the building blocks for more effective and data-driven drug development for the community of drug discoverers and drug developers at large. Next, please. On the front end are our target discovery programs, starting with the Accelerating Medicines Partnership for Alzheimer's Disease, Target Discovery and Preclinical Validation Project. So this public-private partnership is a very large-scale team science effort that uses a systems biology approach to discover a novel therapeutic targets and to validate these for downstream translation. And the program is deploying this open science research model that enables very rapid sharing of data, methods, and results. Next slide, please. Now, since its launch, we have initiated a whole suite of AMP-AD affiliated consortia and programs that are using that model of integrating epidemiologic, genomic sciences, and mechanistic research to gain the very needed deep mechanistic insight on the complex biology of Alzheimer's, its relationship to brain aging and aging across multiple organ systems. And so these programs are pushing out these mechanistic insights and, of course, new insights into targets and biomarkers. Next slide, please. All the data and analytical outputs coming from these programs are being shared through a centralized and integrated FAIR data infrastructure, the AD knowledge portal. And the wealth of data, methods, and analytical outputs has been widely used by the academic community of researchers in industry. In fact, 40% of the users come from industry and pharma. Next slide, please. The output in the form of target nominations, along with all the associated supporting evidence and extensive drugability information, is being shared through the open source platform Agora, where currently there are over 500 candidate targets. Next slide, please. So that wealth of target output is being then de-risked for drug development through our Treat AD centers. So these two multi-component centers are developing open source target enabling tools, reagents, and robust interrogation of the biology of targets, or as seeds for drug discovery. And this is a recently launched program, and they will be providing all that output open to the academic and industry and pharma community. They will also take a subset of these targets, prioritize targets, and take developing drug discovery campaigns around them. Next slide, please. A key tool for therapy development are good animal models. So our Model AD consortium, composed of 2 centers, is developing the next generation animal models for late onset AD. And they're conducting a very extensive, deep multimodal phenotyping of the most promising models and aligning those phenotypes vis-a-vis various aspects of the human disease. And all that model output, the associated data is being shared rapidly and broadly with the community without any restrictions for using basic research or therapy development. Next slide, please. So to date, there's been over more than 50 models generated, and they're currently available. And this is just to show that they're being taken up by academic labs as well as labs from industry and pharma, increasingly so. Next slide, please. So the Model AD has a very extensive preclinical testing core, and the PTC has developed a pipeline, very robust pipeline for rigorous preclinical efficacy testing, where the new animal models can be paired with the most promising compounds based on alignment of mechanism of action. This pipeline is now open to the external to Model AD community. And on the next slide, you can see how you can nominate your compounds to be taken up and for very extensive preclinical efficacy testing by the Model AD PTC. So these nominations are now accepted through the Stop AD portal. The next slide. So all these programs were developed based on recommendations from the NIH Alzheimer's Research Summits. These are triannual strategic planning meetings. Just a plug for the upcoming 4th Research Summit. It is coming up in April to a Zoom near you, and hope that you will be interested to join us throughout that week. On the next slide. Just acknowledging many colleagues in the Division of Neuroscience, particularly our translational research team led by Lara Falla. Thank you.
Thank you very much. That was great. And next, switching to our NINDS colleagues, Chuck Cywin.
Hi, thank you very much for giving me the opportunity to talk about the projects for at NINDS and the Division of Translational Research for small molecules and biologics. Next slide. So we start with a program called Ignite. Ignite is a feeder program. It's designed to get you the pieces you need to start a drug discovery project in earnest. So we have a PAR that's designed to develop the compound and do in vivo efficacy studies. We have a funding opportunity that's designed the assay, do the assay development and screen and identify hits. And then we have a program that allows you to develop and validate model systems or pharmacodynamic markers to facilitate neurotherapeutic discovery. All of these are milestone-driven grants that are designed to get you ready to enter a couple of the other programs we're going to talk about in just a second, either for small molecules or biologics. The last due date was just passed. However, there is an intent to reissue these FOAs, and Becky Roof is the contact at NINDS for these. Next slide. My day job and what I focus most of my attention on is the Blueprint Neurotherapeutics Network. It's designed to help with small molecule drug discovery and development in disorders of the nervous system. It's a program for both academics and small businesses. It provides grant funding to support this all the way to the clinic and to first-in-human experience. The projects can enter on the pathway along the spectrum from discovery stage to optimize a well-validated hit through medicinal chemistry, or if you really have the candidate, you're going to the development stage. It can be things like IND enabling talks or even dose range finding talks, and additionally finishing out efficacy studies and so forth to get ready for clinical testing and filing IND prior. What's really unique about this is we have access and we provide access to NIH-funded consultants, contract research organizations, and expert NIH managers and science officers. We do this in a fashion that allows the projects to move forward at a very good pace. Next slide. There are 9 ICs that are signed on to this, and if your project falls into the mission of one of these 9 neurotherapeutic based ICs, we can certainly take the project in. Next slide. As I mentioned, it's a very interactive program. We have the discovery teams, the development teams that consist of the principal investigator, seasoned industry consultants. These are people who have been at pharma for many, many years and have retired and now doing consulting, in addition to NIH staff. And what we do is we take advantage of the knowledge in the disease biology area and help augment that with whatever else is needed. So we have contracts for medicinal chemistry, which includes in vitro ADME and computational chemistry. We have contracts for PK and as well as tox. We have drug manufacturing formulation contracts. We have clinical trial contracts. And if you're doing drug discovery and development, we have a relational database to help with that. So we can really put together a unique team for you that augments what you need and move it forward. What's really important about this program is that all of the contracts that deal with IP, things like the medicinal chemistry and the drug manufacturing formulation contracts, as well as the consultants, have the intellectual property assigned in either by a separate IP agreement or by language in the contract to your institution. So therefore, you're getting non-dilutive funding, you're getting the intellectual property. The NIH staff, although we're all drug discovery experts as well, we're really facilitators. We don't get involved in trying to do inventive steps on this, and we try to move projects forward. We've had a reasonable amount of success with this program over the last 10 years, many licensing deals. Several projects have been into the clinic, and including a couple now that I think are in phase 3. So it has been a successful paradigm for licensing out and finding opportunities for investors to take projects out of this program. Next slide. What's important on this slide just to realize is we have both a standard track, the UG3 UH3 track for all comers. That would include small business and academics, where the small businesses would be mainly folks that don't qualify for SBIR grants. And then we have the SBIR track for those small businesses that do qualify for SBIR. If you have any questions, you can certainly contact me anytime. I think if you click, there's one piece of automation here, which is just to remind me to say that not all of those 9 ICs will accept a development project. NIDA and NIMH, for instance, will not. And so they'll only take a discovery project in. They both, I think, have additional funding opportunities for those kind of projects. Next slide. I also have responsibility for the non-addictive analgesic therapeutic development program, both small molecules and biologics to treat pain under the HEAL initiative. So this is all about pain. It's very much modeled after the Blueprint Neurotherapeutics Network, and so it's going to have similar kinds of contract support, identical for small molecules. We're still in the process of establishing some of the contracts for the biologic space. and hope to have that in place for FY22. But there is one at least additional feature in this program that's not in BPN, which is the identification of a companion target engagement biomarker is an important component in this program, and we help you do that through funding. The goal of this is to get projects all the way through phase 1 and have them ready to hopefully enter EPICNet, which is our phase 2 program down the road. It just like BPN, it has access to consultants, but you can also work with some of our intramural colleagues at NCATS if you wish to move the projects forward. You would do that by creating a CRADA agreement with them in advance to work it with them. All right, and so the last program I want to just touch on, next slide, is the Create Bio program. And the Create Bio program is for biologics, much like the blueprint was for small molecules. This program is for biologics. It has 2 tracks for optimization as well as for development track to move the project IND enabling in clinical trials. Again, it's a cooperative agreement. It's not, it doesn't have the contract and consultant support. It is a standalone project. I can say in the future that we have a concept to move biologics to the BPN platform as well. So we may stay tuned for that next year. And I think I'll end on that.
Hi, yeah, this is Nick Langhals. Similarly, as I mentioned, I'm at NINDS, and I oversee several programs both here as well as within the Brain Initiative, the SPARK program, and we have a new Blueprint program that's recently been approved as well. Next slide. Generally wanted to make sure and highlight, just so that everybody's aware, that NINDS does cover a broad array of various different neurological disorders and stroke, hundreds of them in actuality. Many of these things are within our mission, and we have targeted opportunities for some of the programs and areas, but even if they're not, we still welcome all comers into our various different programs. Next slide. And so here's a general overview of some of the various different activities that we support. You heard about several of these in Chuck's presentation just before this, but also just trying to highlight that that we have all these opportunities available to support these activities, both in the earlier stage translational, and there's 2 more animations to click on this one. Sorry about that. As well as clinical trials, infrastructure and resources when you get to that stage of your development to support those activities. And NINDS plays lead roles in several other programs, as noted within the Brain Initiative, the HEAL Initiative, and the Common Fund SPARK program. Next slide. And animation too. Yeah, my day job, as I mentioned, is overseeing NINDS's Translational Neural Devices program. There's a couple funding opportunity RFAs as noted here at the top. Similarly, our last due date just passed this week, but NINDS Council approved reissue of these funding opportunities, and we're in the process of getting them back live and on the street going forward. But the overall goal here really is to help try and support that late stage preclinical development with these fast track phased awards that support IDE enabling studies in the first phase of the award, or activities needed for IRB approval for non-significant risk studies, as well as that subsequent clinical trial in the second phase of the project, all bundled together within one award mechanism with an administrative programmatic review at that transition point, assuming that the regulatory approvals are all in place at the end of the first phase. If you have any ideas or thoughts or potential proposal submissions, we recommend that you reach out to us well in advance so that we can ensure that the program is a fit, as well as give you feedback on any of the specifics related to that. And we're welcome to do that, and want to make sure that we find the best target for your project. Next slide. As I mentioned, we cover a broad array of various different neurological disorders in both our NINDS programs as well as our Brain Initiative programs, and we support all sorts of various different device modalities, both invasive and implantable tools, as well as non-invasive strategies to try and alleviate neurological disorders, and diagnostic devices are also included within the mission of several of our programs. Next slide. In my second job as a team lead within the Brain Initiative, NINDS is one of the players across many other institutes, as you see in the list here, where the goal of the Brain Initiative really is to try and develop and apply new tools for understanding how neural circuits underlie complex behaviors, both in health and in disease. And you can click through the 3 animations on this one. And really what we're trying to do here is leverage these technological innovations to enable new discoveries about neural circuit function, use these discoveries as a foundation for future new therapeutic strategies for human brain disorders, as well as then disseminate and democratize these technologies for both basic discovery applications, as well as for ultimate future clinical applications for some of the various tools. Next slide. And you can click through the animations here. Overall, we're in, the Brain Initiative was initially approved over 5 years ago now. There's an overall report that you can see on our Brain Initiative website about what the overall strategy for Brain for 2025 was. Following that, there was a follow-up where we had the advisory committee to the director was assembled to evaluate where we stood within the overall Brain Initiative, how we were doing in our programs, and what some future opportunities might be. And you can read through the details of that. But the key here is that overall, the suggestions and advice were to stay on this productive path that we've already started, and continuing support for these technology development and targeted study of various different brain circuits, to balance investigator-initiated research with team science, as well as to try and devote some additional Brain Initiative resources to transformative projects, combined with ensuring we're enhancing integration with neuroethics, and as well as broader societal implications of a lot of the tools and technologies that we're developing. Next slide. Here's the overall funding, both up until this year for the Brain Initiative, as well as the projected funding going forward, where we've reached or exceeded our steady state projections of about $500 million per year dedicated for funding these types of projects. And there is a big boost in 2023 to facilitate some of these larger scale and launching those transformative projects. Next slide. Oh, yeah, there we go. Next. So far, we have been brainstorming and taking some of the advice of the working group on transformative project ideas. You can click through the 3, I guess, animations on this too. And so you can see the list here of the ones that so far have been green lighted by the Brain Initiative. The first is on organizing resources for brain cell type access and manipulation across species, also referred to as the cell type specific armamentarium, which is really focused on trying to develop these cell type access strategies, not only in rodents and non-human primates, but as well as trying to broaden some of those for human cells and tissue, with the long-term goal of developing new cell-specific target therapeutic strategies for human brain disorders. The first RFA is issued in live, probably not going to make the first due date, but there's a follow-up in October. The second transformative project is continuing our phase 3 development of our brain cell census. So really trying to figure out the parts list of the cell within the mouse brain was in the first phase, and we're going to shift an emphasis to construct a more human-based cell atlas. And then the third is looking at microconnectivity analyses and developing a wiring diagram for the brain across multiple scales and spatial levels. Next slide. So those were beyond the transformative projects. We have all sorts of funding opportunities that cover the broad array of technologies and the technology development and application within the Brain Initiative, that all of our funding opportunities can be seen at the Brain Initiative website listed there. Next slide. And we can click through the few animations here. Similarly, within our team, we have a pipeline of various different tool development, funding opportunities from early concepts to tool development, optimization, and then ultimately for resource dissemination to end users for those various different projects. And the companion R01 focused on understanding the biology and biophysics of how these various different recording and stimulation technologies actually interface with the brain. Next slide. And we can click through the three animations. And so similarly, there are along our pipeline, funding opportunities focused on the human translation that are similar to our NINDS translational devices program. And in particular, these within the Brain Initiative are accept applications are really trying to facilitate invasive tools and technologies, both for diagnostic and therapeutic applications. And we have a companion opportunity for small business investigators. Next slide. And as part of that, we have stood up a public-private partnership program. You can see there's several companies already participating with the goal of really trying to facilitate collaborations between academic investigators and commercial partners in these sorts of class 3 implantable medical devices space for neurological applications. If you're interested in participating, feel free to reach out and we can discuss whether or not your technology is a good fit. Next slide. Second to last here, so the Brain Initiative Investigators meeting is coming up. You can see the website there, brainmeeting2021.com. While this is an excellent opportunity, if you'd like to figure out and understand more about what we're funding within the Brain Initiative and potential opportunities to collaborate with Brain Initiative investigators or looking at and exploring licensing opportunities, the meeting is open to the public and registration is available now. Will be a virtual meeting in June this year. Next slide. And so to summarize, really just emphasizing that we do have a full pipeline of opportunities, both within NINDS and one animation here to click, as well as within the Brain Initiative to support technology development all the way from concept generation through clinical trials. And with most of these programs, as noted on there, some of them have expired, but reissues have all been approved by council last month and should be forthcoming new versions in the very near future. Thanks.
Great, thank you very much. So again, if you have any questions, please use the Q&A. We have a few that have come in. The first, getting to the heart of the geroscience hypothesis, the question is, when does age become a, quote, disease, end quote? And of course, the question there is, does it become a disease? So Ron, we'll start with you and then open to anyone else that wants to add in.
Sure. If you look at the mission statement or strategic goals for the NIA, one of the opening lines is aging is not a disease. And it isn't. And from the point of view of the FDA, it's not a disease because it happens to everyone, unless you're unfortunate to die young. So the point about the geroscience is that aging is a risk factor for loss of function, for advent of disease, and for also for disparities. We have to keep in mind that there are health disparities that where aging is a critical feature. And so there are things that happen over the life course that will set the stage for when aging is evident as opposed to when aging begins. They're two different questions. Aging might begin very early in life, but it might be that it's not aging per se, but how well an organism develops and how much function and functionality it has. And then as systems begin to fail, buffering of systems fails, there's a decline. And that's the part where at least my division tries to play a part. But to call aging a risk factor means that you have to approach it from 2 points of view, because the risk factor is dose and duration. So what is the dose of aging? In a sense, it's how much a system has begun to fail, and the duration is pretty much when has it begun to fail. So I think that would tell you then that you would not try to intervene in aging in a developing organism, because you are taking a system that is biology before it fails. You might want to be able to get the system to its optimum and then deal with preventing failures after that point, or delaying them or ameliorating them. So then there are two aspects to that. One is loss of function, which seems to be kind of linear, and therefore could be explained by single causes, although it's unlikely because aging is complex. And the other is the advent of diseases is actually exponential. That exponential feature means you have at least two factors interacting. Even if those factors change linearly, the interaction would give you an exponential outcome, or some form of exponential outcome. So that's the surprisingly short answer for a surprisingly complex question.
Anyone else want to weigh in on that?
So I'll add to that as well. And a very valuable answer from Ron. Within the Intramural research program, there's a very strong interest in healthy aging and resilience in aging. And both from a basic point of view in across various animal models, that's run by my branch chief, Rafa De Cabo, as well as the clinical perspective, which is run by Luigi Ferrucci in the Baltimore Longitudinal Study of Aging, which is a hugely powerful program that looks at healthy aging across the US population, and has a whole series of biomarkers that it's looking at. So it's a very important area, and it's a very important area for healthy aging to improve longevity and reduce disease burden in the United States. A vast area that needs substantial input from many corners, including commercial input as well.
Great. Anyone else?
Hi, Todd. Just to amplify in support of what Ron and Nigel said. So the Division of Neuroscience, we invest in many ways in supporting research to look at that dynamic interaction between brain aging, neurodegeneration, but in the context of aging. And in part, we're doing that by these systems approaches where we're bringing the wealth of epidemiologic research insights, longitudinal multi-omic data, and marrying that through data science with mechanistic research. But it's dynamic, and it has so much potential for revealing points of intervention, particularly in that resilience space. We, of course, like the other divisions, support research on understanding the molecular determinants of resilience and resistance to different types of AD risk, but also in the context, again, of aging. Huge implications for disease prevention.
Expanding on something that a couple of you just touched on, in terms of animal models, could each of you speak to animal models in general? That's something we hear from our companies a lot in terms of a lack of truly predictive, clinically predictive animal models, and how each of the programs in that, preclinical development, late preclinical development of therapeutics handles either the lack of predictive clinically predictive animal models, or incorporates ones that you do find predictive. I'd be interested in everyone's opinion on this. Why don't we start with Chuck?
Maybe I can start, Todd. And
go ahead.
You can follow, since I shared about our Model AD consortium. So that's one of our big ways to actually remedy some of the major issues that plague, that preclinical to clinical translational gap vis-a-vis animal models. And one is that the first set of models were for late onset risk factor genes. And again, models are not models of disease, they're in a way models of targets or biological processes. So this next generation that are developed with leveraging the genetic risk factor insights and the predictions from the systems biology efforts. So this next generation of animal models, one of the ways by which we're working to make them more predictive and translatable as tools, is to do this multimodal phenotyping both across domains, whether it's omics, biochemistry, imaging, other features relevant to human outcomes, but also across aging trajectory. So these most promising models, those that have molecular homology with AD signatures, are then being phenotyped all the way through 24 months. And so all that data, RNA-seq, proteomic, biochemical measurements, is being made available, and so that we can understand and pair, right? If you're going to take a model, you want to prosecute a pathway, an intervention for a particular mechanism, you can actually have a collection, suite of models to choose from and choose the most appropriate one, where you can use actually as a PD model of targets. You can actually, and part of one of the things that we are embedding here is particularly capturing peripheral signatures, proteomic and metabolomic, building that into the phenotyping and with the potential to use them as translatable biomarkers.
Perfect. Others?
I would say at NINDS, for my programs, the animal models play a role, but they're not always definitive in terms of what's going on, because many of them are not predictive. So we do look at it on a case-by-case basis, and we're looking why target engagement biomarkers are really important, because then you can potentially get a read, are you engaging that target? You may not have, that particular model may not be the best model for predicting translation, but if you can show that you've engaged the target, and when you get to the clinic, you'll at least know that you can show that you have target engagement, and you can test hypothesis in humans. We do have, places where the models have been more predictive. Things like the epilepsy therapy screening program that we have at NINDS has been a long-standing program that has been utilized by many people over the years and has resulted in a number of drugs and has been part of their development pipeline. We also have the pre-screening platform for pain that's being stood up again, designed to do robust assays to the community in the pain field in there. And you can think of for the HEAL, the Helping to End Addiction Long-Term program, which is again focusing on non-addictive treatments. The addiction models have been quite predictive, and we again would expect all projects to go through there to be utilizing things like the self-administration model and stuff to show the addiction liability of their programs.
Yeah, so to add to those really excellent answers, as was pointed out, they're models. They have caveats and they have their advantages as well. And so one of the important things is to use models so that you can cross-validate each part of the drug development process, and to use them in such a way, as was pointed out by Chuck and Suzana, so that you can actually develop biomarkers, whatever they are, preclinically, that you can then look at and test in humans, so that you can then cross-validate many of those aspects. And so the animal models can be used in many ways. And but to set up a rational way of developing your device or your drug through animal models at the very beginning is very important, and helps you practice those techniques when you actually get into the clinic, and when you cannot repeat those clinical studies because they're so important. So you really have to optimize those studies and maximize your potential to be successful there. And animal models can help you do that as well.
Great. And we have a couple of follow-up questions that are coming in on this, and then we'll move to other topics. And the first is about the lack about the role organoids can play, in terms of providing another model. And the question being about what context of use and validation data would be required before we see organoids more widely adopted. And if anyone wants to speak to how you see organoids currently being adopted, I think that would be helpful as well.
In a somewhat speculative way, we do know that organoids can often recapitulate some features of the live animal physiology of the whole animal. One good example would be organoids created from intestinal stem cells, which have a lot of the features, even though they don't look like an intestine with all the folds, and they come out in little balls, they have the same lineage patterns and budding off as you would see in an actual intestine. One of the issues, though, vis-a-vis aging, is how do you create an organoid that recapitulates age of the animal? And there's some progress that's been made on how to, say, take a cell and induce pluripotency without resetting the entire epigenome so that you have an aged epigenome. There's also some very interesting work coming out on organs on chips and integrated organs on chips. So these are going to be very good tools, and you can, since they're cells in culture, you can engineer them for specific characteristics, including reporters of different features that you might like to screen drugs for. So it's, you know, it's high tech, but it's a great idea. We'd like to see more of it.
And the other part of this question that came through was also specifically around blood-brain barrier and actually assays that can, you know, use human brain cells and measure blood-brain barrier transmission. And you know what's stopping that kind of adoption. But I think actually some of the stuff that Ron said actually can pertain in that way as well. And validation and other ways that we see these being adopted in research studies, I think then only further the use. And that's what we see. Anyone else want to add to that? No, great. So Nigel, do you want to say something?
Yeah, I mean, I think it's part of the jigsaw puzzle, and one has to combine many of these things together. I think in addition to what's already been explained, we now know that certainly for brain, you know, for neurological disorders, it's not a single cell type. It's that the cells in the brain interact together, and different cell types interact together in certain ways to provide a final response. And in cell culture, you can look at cells singly, or you can put them together. And in organoids, this is another more complex way of how the brain interacts. So to have those models available is going to be hugely useful as they develop in the future, and they'll be applied more and more.
Great. And with the last few minutes, we have a couple of questions that we'll try to hit quickly. One is from someone that's from an engineering platform, developed a platform that monitors engagement, and was interested in kind of who they would contact to see if there's interest in technology across various research areas, to see if there's an impact on engagement and quality of life. So I think the type of engagement that they're monitoring may not be relevant to these specific programs, although, you know, any panelists, please correct me. But, you know, I think some of our interests, it definitely relates to some of our interests from our division of behavioral and social research, and also from our office. So please feel free to get in touch with us on that. There was also a question about remote patient monitoring, and in terms of Parkinson's in this case, and looking at disruptive technologies within remote patient monitoring, and a question of what interest there is from our institutes to fund clinical studies of remote patient monitoring that could then support CMS to consider CPT codes. And I'm not sure if anyone knows of any specific interest or funding opportunity that they want to speak to there.
So Todd, just on that, we don't have anything specific. Certainly the question was about small business piece. If I remember the question correctly, obviously our small business funding initiatives support clinical research or in clinical and early stage clinical trials, but we don't have anything specific for remote patient monitoring. Also, we should say that Parkinson's disease, we do have a portfolio on Parkinson's, but it's fairly small compared to the very large, extensive portfolio that NINDS has, as that's Parkinson's is in large the purview of our sister institute. So maybe our colleagues there have
And yeah, I guess from the NINDS perspective, as Todd noted, most of our funding opportunities we talked about today were focused on kind of new technology development and translation. But NINDS does have a funding opportunity that it accepts applications looking at comparative effectiveness research in the clinical neurosciences. So kind of depending upon the study design and specifics, that could be a potential option. And we're happy to put you in contact with the program director that oversees that program.
Great. And I see that we are just out of time. Maybe I'll throw in one final question and see if anyone wants to address this real quickly. And that's that looking at neuroethics and the need to broaden neuroethics to include behavioral lifestyle aspects beyond medicine and technology currently typically addressed. So does anyone have anything that they want to say in terms of incorporation of aspects that we can raise quality of life? And it's, I don't know if you've seen this, Nick, in any of the kind of brain programs, if this has come up.
Not directly, but we do support research on kind of the ethical implications of the advancements in neurotechnology and brain science. So there is an R01 funding opportunity within the Brain Initiative to support some of those activities. And the other potential home for those types of studies could be NCCIH, the National Center for Complementary and Integrative Health, that does fund various different research projects looking at alternative and integrative health strategies for improving overall patient care and well-being.
Great. Well, I want to thank each of our panelists very much. This was a great conversation. To Suzana's answer just previously, yes, we do have, of course, both NINDS and NIA, several funding opportunities for small businesses, majority of which are open-ended, are investigator initiated. So things like the RPM and some of the other questions that have come through, we definitely would be interested in talking to you. And I just put in the chat box how you can reach us if you would like to talk about a potential application. Thank you to everyone. Thank you to the panelists. Thank you to attendees. And look forward to seeing you at our future sessions today. Take care.